Everybody. Beautiful morning. This is today's CME code, which is now working again. So the North Carolina Neurology Society is now accepting registration for their event on February 23rd, which is in Greensboro, which is about an hour from here at that nice little resort they have right off the road there. So I remember when it was there last time, and we've had an increasing presence at these meetings, and I think that's great. I think it's a great organization, and we should have as many posters as possible at it. And Joel is now one of the leaders of the society, so try to put it in your schedule, February 23rd to 25th. Amongst our all-stars, Danny Laskowitz and Luke James helped mentor Gabrielle, our neuro ICU fellow and future faculty member, to get a fellowship through the Don Sanders Research Fund to help him pursue his research on vestibular stimulation in patients with a minimally conscious state. So congratulations, and coffee cups on the way. Wayne Feng wrote a report on how cancer affects stroke risk and about a trial that he's doing with our UNC colleagues to look at that. Nandini Aburi, who's a hospitalist here and at Duke Regional, on why she loves being a neurologist. And she's talking about how it's important to experience your patients. It's more than just the disease the patient has, it's the patient who has the disease, and being aware of that and using that to form a good bond. So good for her. For the older folks, including myself, it's open enrollment for your benefits. And that's true for you, Chala. By October 27th, if you want to make any changes in your benefits, you have to do it. Today in history, today is National Fine Arts Appreciation Day, which, it's not for me, but Greasy Food Day for sure, and World Pasta Day. And today, Will found out that is the day that the first microwaves went on sale in the US in 1955. But before that, in high school, remember, I don't know if you remember, we used to put nails in the hot dog and pass electricity through it and cook it that way. It was kind of like your own version. In this one, they were using the fork there. So microwaves in 1955 sold for approximately $13,000 because they were so fancy. And they probably were magical, not as magical as when potatoes came out of a box. That was, you folks are all too young to remember that, but when mashed potatoes came out of a box, that was a revolution. It wasn't expensive. Brian McCrory was part of a new study looking at outcome of stroke patients in the various different stroke centers, and found that the highest level, the thrombectomy capable stroke centers, led to a better outcome. Well, an expected but nice finding. Laurie Sanders was part of a team that used a new CRISPR system from Staph aureus called SADCAS9. I was looking at that last night, and I thought it was Staph aureus and then a D for something, to look at a controlling expression of alpha-synuclein. So good for her, and nice name. So I started working on Did You Know, and I just got way too complicated, so I'll have it next week. It's like, what is a county? And why do we have a Durham County and a Durham City? And why it's not clear what do what? And are those people called sheriffs really police officers? So we'll have that. And the answer to that is yes. So in Boston, the sheriff just takes people from prison to court. Down here, Durham County sheriffs are the actual police of Durham County. So be respectful. Again, today's CME code, and today's case presentation is Dr. Austin Peters. So Austin, come on up. Ooh, that's Chala. Is that for me or for Austin?
For you, Rich.
Thank you. You know the answer. I stop share. And then share. And then hide. Ooh. All right, take it away.
All right. My name is Austin Peters. I'm Child Neurology PGY3. So thanks for having me here to give this case presentation today. It's a somewhat long case, so I'll move pretty quickly through it, just trying to highlight the key details. So we had a 61-year-old male who had a past medical history of paroxysmal AFib multiple myeloma. He had a stem cell transplant earlier this year. He was continued on maintenance chemotherapy. He had a more remote history of an oligodendroglioma that he had resected and chemotherapy for back in 2017. He had developed seizures around that time. And then more importantly, he had recently developed a right buttock abscess. He presented to us with one day of double vision on primary gaze that resolved when looking to his left. But to fill you in a little bit on what had happened in the week prior to him presenting to our emergency department, he had what he was told was a lipoma that became infected in his lower back. He was then started on empiric Bactrim. He then re-presented to the ED one day later, and they did a bedside IND because they were concerned it had become an abscess. And his Bactrim was then switched to doxycycline because in the interval period, he had developed an AKI. Then he re-presented a day after that with acute onset of right foot pain and swelling, and he was diagnosed with gouty arthropathy and then discharged again. And so his exam for us, it was notable really only for this restriction of right lateral gaze in the right eye that was consistent with a right CN6 palsy. So you can see in this topmost picture, he's attempting to look to the right. On a few of his other attempts, he was able to get that left eye more medial, but the right eye, he could never do better than this. And then you can see he has a very normal gaze at midline, and then he does quite well when looking to the left in that bottom picture. His wound culture of note had, from that abscess that was IND'd, had grown positive for nocardia about a day prior to his presentation. And then he had a CT brain in the ED that was unremarkable. And then his labs, just a few of them that were collected, his ESR was elevated at 58, his CRP was elevated at 6.1. Although it was difficult to interpret, one, in the infection of his known abscess that he had. And because all of the previous data we had for his ESR and CRP were from several months ago, closer to the time of his stem cell transplant, and every time that he'd had those labs collected, they were always elevated. Of note, he did not have a leukocytosis. His white count was 6.0. He was afebrile. He had 2 blood cultures drawn that were negative. But he did get an MRI brain with and without contrast, and it identified this 10 mm right dorsal pontine lesion that was described as a classic figure of 8 appearance that was consistent with CNS nocardia. I think if you maybe squint, you could convince yourself that maybe there's an 8 there. There are a few other lesions that I haven't included here. He had other enhancing lesions in the left cerebellum and the right parietal deep gray matter. And then he did have a right frontal semi-ovale and left occipital lobe infarcts on diffusion that were concerning for septic emboli. And here is the right frontal semi-ovale one. And then you can see on the other side where he had that oligodendroglioma that was resected. And so he's had a very long clinical course. He's still in the hospital today, but just to highlight a few of the key points from it, he was initially started on empiric double coverage with Bactrim and imipenem. He initially had a TTE that was negative. So then he had a TEE that was also negative, showing no signs of endocarditis. At one point shortly into his course, he developed acute onset right facial droop, left-sided numbness and paresthesias and focal facial twitching. He had an EEG done. He didn't have any evidence of seizures, but there was concern that he was having worsening of his infection or perhaps just edema from the known infection he had. So he was actually brought into triple coverage with linazolid as well, and then Decadron was added. After about a week in the hospital, his culture did speciate as nocardia pseudobrasiliensis, and he was switched then from imipenem to moxifloxacin based on some previous Duke culture data from other patients who had this same bacteria growing. And so to talk just very briefly about nocardia, it is an aerobic actinomycete. It is a gram-positive bacillus. You can see in this picture in the upper right-hand corner, it has a very strange kind of branching filamentous structure, almost like hyphae in appearance. It's a pretty rare infection, only about 500 to 1,000 cases a year. It does mostly affect immunocompromised individuals like our patient was, but the literature does describe roughly a third of patients who have a nocardia infection were previously healthy. So it's important to consider even patients who are not immunocompromised. And then just to very briefly touch on it, it is a ubiquitous bacteria. It's in soil and the waterways everywhere. And so the thought is that people who have either are immunocompromised or have pre-existing pulmonary disease will inhale some of those spores, and that's how they end up with pulmonary nocardia. But the patients who have cutaneous nocardia like ours, usually the thought is that they have some sort of traumatic event that introduces it, although our patient, interestingly, did not ever report any trauma to the area where he had the abscess. Then to talk very briefly about CNS nocardia, It is a diagnosis where 90% of patients or more have a ring-enhancing lesion, like ours did on MRI. Most people will have pulmonary or cutaneous disease, like our patient did. However, about a third of patients present with what's termed primary CNS nocardia. So they have no other evidence of a nocardia infection. Of course, the culture is the definitive way to diagnose it, which is really challenging if someone only has CNS nocardia, because then it becomes a serious risk versus benefit discussion about whether or not it's worth trying to culture these CNS lesions to guide your antibiotic therapy. Just to talk very briefly about some of the presenting symptoms. About half of patients will have focal neurologic abnormalities like ours did with his CN6 palsy. Slightly under half have headache, about 40% have fevers, and about a third have seizures. There is a very rarely described meningitis when someone has a more disseminated CNS nocardia infection. And then to just touch on the treatment, there is very limited high quality evidence for this. There aren't any RTCs. It's really based on our sensitivity data and case series. Most species are sensitive to Bactrim. The treatment, of course, whenever possible, should be guided by culture. And the duration, unfortunately, is very long. It's 6 to 12 months based on clinical and imaging response to treatment. And so here I have, just from UpToDate, pulled out some of the empiric regimens. Our patient was started essentially on this top one, where you're on Bactrim and imipenem. You could argue that he had CNS plus cutaneous disease, so maybe he should have initially been started on triple therapy, which he eventually was on. And then to kind of sum things up, the prognosis, unfortunately, is still 20% for all-cause mortality at 12 months in these patients. However, it's important to realize that there was at least one case series that showed that about 89% of patients who were promptly treated did end up having, were cured from their nocardia infection. So it just kind of highlights the importance of keeping this on your differential, especially early on, to try to get empiric coverage. And our patient, to bring it back to the case, he did ultimately have a follow-up MRI at 3 weeks into his hospitalization. And that MRI thankfully did show improvement of his CNS infection. At this point, his expected treatment course is at least 6 months. And it's an open question whether after he finishes his treatment course, if he'll need kind of ongoing lifelong prophylaxis against future nocardia infections. Thank you.
All right, great. Thank you. Oh, yeah. Chris, that's a little bit of a thing, huh? Try plugging it into its other. Oh, but it's a.
I think it's going to go
Think
in
it'll
one
be?
of those. Yeah. Yep.
Oh.
Yeah.
Good. All right, everyone. Welcome back. I would say today's audiovisual challenges have been relatively manageable. We're really happy to have Cagla Eroglu here today. When I was young, everyone just studied neurons because they thought those were the cells that mattered in the brain. So Cagla is one of the reasons why that theory was put to bed for quite a while. And now the neurons are, thought of more as just like the wires that bind the glia together, and the glia do like all the real work. She is the vice chair for research in the Department of Cell Biology. She's a professor of neurobiology and a Howard Hughes medical investigator. Cagla is a superstar. She got her PhD at the European Molecular Biology Lab in Heidelberg, and then did a postdoc with the great Ben Barres at Stanford before she came to Duke. At Duke, her work on the role of astrocytes, glia, microglia, in both maintaining normal cellular function and in leading to neurodegenerative disease has made her one of the top neuroscience researchers in the country or in the world. And we're really thrilled to have her come here today and tell us about her work. So Cagla, I will turn it. We're really happy to have you here. And I'm sure this is your first time over in this neck of the woods. Well, welcome. So let's do share screen. This looks exciting. Floating. I mean, I'll just tell you how old I am. When I was a graduate student, with Ben Barris, who was eventually became Cagla's postdoc advisor. So that's how long I've been around, and that's how young and successful Cagla is. Okay, so I think, fingers crossed, we're okay.
Can you switch to it's in presenter mode?
Yeah, can you text it to me? Because we can't hear what you're saying. Are we on mute? Maybe that's what he's saying.
Oh,
Let me see.
doesn't look like we're on mute.
So can you
Yeah.
hear us out there?
Yes, we can hear you. It's just on
Okay.
presenter
All right,
mode, so we
we're
can't see the
I don't know what
slide.
Will was trying to say. All right. Go ahead, Cagla.
Thank you so much, Rich, for that extremely kind and super generous introduction. And I'm really happy to be here today. And I've had the pleasure of interacting with Rich over the years. Has been always super generous with his praise and support. And I appreciate that a lot. So as Rich alluded to today, I'm going to talk about the other cells of the brain most of the time. However, I will talk about how they interact with neurons. And don't get me wrong, neurons are super important. Nobody's arguing against that. But I think we are now at a stage in neurobiology and neurology to understand that the brain is a whole organ, which is composed of many different cell types, and how it's wired is also regulated by all those cell types and not just neurons. So let's hope we can progress here. Let's see. All right. Okay, so in my lab, we study a fundamental question in neuroscience, which is studied by many. And it's basically to understand how brain circuits are formed during development, how they are maintained and remodeled throughout our lives, and what happens if these developmental and lifelong processes of circuit wiring are impaired in the cases of disease. And this is a difficult question that is keeping a lot of us busy because of the sheer complexity of the brain. Human brain contains billions of neurons, which are wired together through these submicron structures we call synapses that are formed between a presynaptic axonal button and a postsynaptic dendritic side. And there are hundreds of trillions of this in the brain. And even more complicated is that these neurons have to, wire through the mesh of many other neurons, find their appropriate targets, and make the appropriate kinds of connections to each other. So, and a very influential hypothesis that guided much of the cellular and molecular work in the field has been that neurons have genetically encoded a cell adhesion and secreted protein-based chemoaffinity code that essentially provides the blueprint to wire brain circuits.
We're getting our colleagues are telling us to use slideshow. Okay.
All right, I hope it's okay now on Zoom. All right. So this cell adhesion and secreted protein-based neuronal chemoaffinity code.
These things that are irritating.
Thank you. And one of the opportunities this influential hypothesis have given us is to figure out how astrocytes project their axons during early development and find their targets. But a lot of other processes are ongoing during the formation of synaptic circuits. And one important one is to find the proper balance between different types of synapses. Not only you have to find the correct partner, but also each neuron has to figure out a way to balance the different types of connections that it wires through. And in the mammalian cortex, the model part of the mouse brain that we study, for example, the visual cortex, there are at least two different kinds of glutamatergic or excitatory synapses that needs to be wired early development. And one is the connections between cortical axons and cortical dendrites, and they need to form these connections in concert with the other glutamatergic connections that are formed between axons that are coming from elsewhere in the brain, such as thalamus, and forming thalamocortical synapses. And it's really important to have a balance between these two types of glutamatergic connections. Otherwise, sensory processing and a lot of the processes that require integration of sensory inputs will be impaired. In addition, I'm sure you've heard of it, there are many interneurons in the cortex, and these interneurons are important to provide inhibition to cortical neurons. Without the proper inhibition, then the brain will get epileptic and there will be seizures and there will be too much excitation, therefore leading to excitotoxicity and many different really huge problems for the brain. So in addition, the interneurons are probably one of the most diverse classes of neurons. There are many different molecular flavors of those. For example, they can be marked with markers like somatostatin or parvalbumin. And these different molecular markers also mean that there are distinct roles for these to provide inhibition to the dendrites of the cortical neurons versus to the cell bodies of the cortical neurons. So how do neurons figure out this balance and form these synapses in an appropriate way so that they are appropriately excited and not overexcited and not over-inhibited? And I will tell you today, because we work on those cells, is that this is not only mediated by neurons or their neuronal chemoaffinity code. There are actually other non-neuronal cells in the brain which are integral parts of synaptic circuits, and these cells are astrocytes. So when we think about synaptic circuits, we're thinking about neurons floating in darkness. But essentially, there is no dark part in the brain. It is covered with a meshwork of another network. It's the network of the astrocytes. Astrocytes are these highly complex cells, which are also sometimes named glia. Glia essentially means glue, and anything that wasn't neuron was named glia in the brain. That's where the name glia comes from. But astrocytes are the most abundant glial cells in the brain, and they have this amazing morphology. They go and infiltrate the surrounding neuropil because they are structurally associated with as many synapses as possible. Here are some numbers. In the mouse, a single cortical astrocyte would interact with around 100,000 different kinds of synapses. And then it also gap junction couples to neighboring astrocytes. And this way it forms the syncytia, which is able to monitor and sense synaptic activity across the entire cortical tissue.
So, John, this one, the astrocyte is the kind of bluish thing?
Yes, astrocyte is the bluish thing, and it's the teal. Yes,
teal.
Absolutely. Absolutely. And this is not just a structural association. I mean, the astrocyte can actually sense what's going on at the synapse because it expresses all sorts of neurotransmitter receptors. In addition, it changes the extracellular space by changing its little structures around the synapse, and therefore control both the concentration and the abundance of all these synaptic modulators. But importantly, work from Barrett's lab and my lab and many others in the last couple of decades have shown that astrocytes, especially during early development, are very important controllers of synapse formation, maturation, and synaptic plasticity. And how do astrocytes do that? And we know that astrocytes do that in at least two manners. And one of them is, remember I was mentioning that chemoaffinity code to you that the neurons establish between each other to find appropriate targets. Turns out work from our lab have identified repeatedly that neuronal chemoaffinity code is an astrocyte neuron chemoaffinity code. And I will go as far as to say it's probably a glia neuron chemoaffinity code. Other glial cells are also manipulating it. Astrocytes go in, interact, and modify the chemoaffinity code to change the excitation inhibition balance in the brain. Here, I will very briefly summarize a large body of work that was done in my lab to proceed and present you an unpublished story at the end. But just to walk you through, one of the first mechanisms that we identified was mediated through a synaptic adhesion complex, which is made between presynaptic neurexins and postsynaptic neuroligands. But this was, until we started to work on this in astrocytes, this was predominantly studied even by us as molecules of neurons that wire neuronal circuits. But we repeatedly realized that these molecules are not restricted to neurons in expression. They're also expressed by astrocytes. For example, astrocytes express neuroligands, which we found is very important to bridge with presynaptic neurexins to regulate the arborization of the astrocytes, so the astrocytes can take that amazing morphology and interact with as many synapses as they can, and B, it regulates the excitatory inhibitory synapse formation within this domain. And this had two important outcomes for us in the field. One is that astrocyte morphology is directly related or correlated with the synapse formation. So as synapses form, astrocytes should arborize as well. If they don't arborize properly, they're interfering with synaptogenesis. And B, the genes that encode for neuroligands are linked to autism, schizophrenia, bipolar disorder, depression. And most of the time when we think of their function, we think about some neuronal dysfunction that is leading to synaptic dysfunction. But in this study, we targeted neuroligands specifically in astrocytes, and we found that that was sufficient to cause synaptic dysfunction, indicating that astrocytes can be the cellular origins of synaptic dysfunction in disease. We went on and used this knowledge to identify a couple of other cell adhesion molecules that are working with the neuronal chemoaffinity code. In a very nice collaboration with my next door neighbor, Scott Sutherling's lab, we used a in vivo proteomic approach and identified neuronal chem or NR-chem as a adhesion complex at the junction between astrocyte processes and inhibitory synapses. And NR-chem was very interesting. If you lose NR-chem, astrocytes actually don't become smaller. The opposite, they become larger and they invade the territory that should be otherwise occupied by GABAergic inhibitory synapses. And what you end up is a brain where inhibition is greatly diminished and excitation is enhanced. And more recently, we targeted HEPA-chem, which is again a syndromic autism gene. And it turns out HEPA-chem is bridging the astrocyte syncytia and making sure that astrocytes make gap junctions with its neighbors and not make gap junctions onto itself. As you can imagine, gap junctions are super sticky. And if you start as a cell couple to yourself, you will lose the ability to couple to your neighbors. And this also greatly changes the strengths of excitatory and inhibitory synapses. But how do astrocytes, through these adhesion molecules, control the excitation-inhibition balance? And that brings me to the other part of the coin, how astrocytes actually control synapse formation and remodeling. And that's through secreted synaptogenic proteins. Previously, work from Ben Barris's lab identified that astrocytes secrete proteins which very strongly control excitatory synaptogenesis, and one of those are the thrombospondin family proteins. And in fact, my early work, first as a postdoc in his lab and later here at Duke, was to identify the molecular mechanism through which thrombospondins regulate the formation of intracortical synapses. Moreover, in my lab, we identified another astrocyte-secreted protein, Spark-like-1 heaven, to be a very important regulator of another type of excitatory synapse, the thalamocortical synapses. And heaven is not only required for the formation, but also for critical period plasticity of thalamocortical connections. But one thing that remained for a very long time unknown is how do astrocyte-secreted proteins control inhibitory synapse formation? And this is a story that I will talk to you about today. And there was a reason for this, a technical bottleneck that stopped us from identifying astrocyte-secreted proteins that induce inhibitory synapse formation. And this bottleneck was solved by a very talented postdoc in my lab who's currently interviewing for jobs and hopefully will start her lab next year. So Dolores came to my lab with a very determined intention to figure out how astrocytes control inhibitory synapse formation. And one of the reasons that we couldn't address this question before was to study the role of astrocytes in synaptogenesis. Ironically, we need cultures of neurons which lack astrocytes, so that we can add the astrocytic molecules separately onto these astrocyte-free cultures and test how they stimulate synaptogenesis. And previous cultures we used that were developed in the Barris lab were retinal ganglion neurons, which are all excitatory, so they will never form inhibitory synapses. But Dolores was able to circumvent this problem by purifying cerebral cortical neurons from neonatal rats, and then also purifying astrocytes, but keeping them separately. And then when she did this, she realized that as it is in the cortex, cerebral cortex, she's able to get 80% excitatory and 20% inhibitory neurons in her mix. And these neurons, these neuronal cultures do not contain astroglia in them, which is great. Now we can take astrocyte conditioned media, or I will call it ACM for the remainder of this talk, and then add it on to these cultures and determine if astrocyte secreted factors can induce synaptogenesis. And first, we'll do this for excitatory synapses, which is something we know that they do. And we can assay the synapses by staining with a presynaptic marker, in this case, bassoon, and a postsynaptic marker, Homer1, which is a marker that's specific to excitatory synapses, and look for apparent colocalization of these two signals. As you can imagine, one is in the axon, one is in the dendrite. The only time they will appear to colocalize is when they are at extreme close proximity at the synapse. So we're using that close proximity as an assay to determine where the synapses are. And when Dolores did this assay, as expected, she found a significant increase in the numbers of excitatory synapses when ACM was given to neurons. But what about inhibitory synapses? And for that, she decided to focus on inhibitory synapses made onto excitatory neurons and stain them with, again, a presynaptic marker bassoon and a postsynaptic marker that's Gefrin, which is specific to inhibitory synapses. And when she did this, she also found a profound increase in the numbers of inhibitory synapses that are made onto excitatory neurons. And she actually did the control and checked if the inhibitory neuron numbers were changing with astrocyte treatment, and there wasn't any change. There was always 80% excitatory and 20% inhibitory neurons in these cultures. So now this gave her a platform to screen for secreted molecules that we know are made by astrocytes and whether they can induce inhibitory synaptogenesis. And she screened a certain number of candidates, and I'm not going to, show you all the different candidates she's studied, but the one that she found to have a profound role in inhibitory synaptogenesis. And it's a protein known as Neurocan, which is a protein that is predominantly made by astrocytes. These are the mRNA expression levels in the human and mouse brains. And an average gene expression here is under 5 FPKM, and this is in astronomical levels in astrocytes, as you can imagine. Other cells also make some of it, but it's predominantly made by astrocytes. And NCAN is a converted sulfate proteoglycan, which has a lot of these sugar moieties in the middle, and it has protein interaction domains on its N and C terminus. And when you stain astrocytes and for the antibodies that are directed towards the N and C terminus of NCAN, you can see that astrocytes make this protein, as well as you can find it in the extracellular matrix because it's secreted. And one of the reasons the reason that NCAN turned out to be a very promising candidate was the results of her screen when she added increasing amounts of Neurocan or NCAN into these astrocyte-free neuron-only cultures. This protein at an increasing dose-dependent manner was capable of inducing inhibitory synaptogenesis in these neuron-only cultures. So this made her to go ahead and target NCAN more broadly in the mice. She decided to make a global knockout, and one of the reasons for this is Neurocan is one of the longest lived proteins found in the mouse brain. Its half-life is 90 days. So to do a conditional or to eliminate it later in life, it was going to be impossible for us. So we went with a global knockout. And as you can see here, wild-type Neurocan, interestingly, is cleaved into two, and it's present in the brain as 2 fragments. And luckily, we have antibodies that recognize both. And from now on, the N terminal will be in magenta and C terminal in teal, so you can hopefully recognize the localization of these different proteins with respect to each other for the remainder of my talk. And in her global knockout that she generated, she saw that there was no Neurocan to be found. Both the N and C terminal fragments were gone. And then how does loss of Neurocan impact? Yeah, I hope
It's
you.
okay, just keep going.
Okay. If we all of a sudden stop, that means we ran out of battery. All right. So, okay. I'll try to be quick. Okay, how does loss of Neurocan then impact inhibitory synapse numbers and function in the mouse cortex? And for this, Dolores focused on a part of the mouse cortex known as anterior cingulate cortex, ACC. ACC is an important part of the brain that regulates a lot of the complex behaviors and mood, and is linked to diseases such as schizophrenia and bipolar disorder. And Neurocan SNPs have been linked to these disorders, so that's why Dolores decided to focus on this part of the brain. And she analyzed the numbers of excitatory and inhibitory synapses here, and I'll just show you the inhibitory synapse numbers for the moment. And she found that indeed the knockouts have reduced numbers of inhibitory synapses. And she could confirm this also by electron microscopy. In electron microscopy, inhibitory synapses are called symmetric synapses, and there is indeed a significant reduction in the numbers of symmetric synapses in the knockout cortices. Moreover, she collaborated with another very talented postdoc in my lab, Dr. Shi Wang, who is an electrophysiologist. She went and recorded from the layer 2-3 excitatory neurons in the wild type and littermate neurocan knockouts and found a profound decrease in the frequency of frequency of inhibitory postsynaptic currents, which is an indicator of lower numbers of inhibitory synapses and inhibitory synaptic dysfunction. So this gave her a lot of promise, as neurocan being an important protein coming from astrocytes regulating inhibition in the cortex. And she decided to determine the molecular mechanism. And for that, she started to investigate first the localization of N and C terminal parts of the ANCAN in the mouse brain. So one of the things that we knew about ANCAN before we started this project is it is known to be a part of a structure in the brain called perineuronal nets. These are chondroitin sulfate proteoglycan-rich extracellular meshes that are formed around the cell bodies of certain types of neurons in the cortex, such as the PB interneurons. And you can stain them with a lectin WFA, and you can see them really nicely surrounding some neurons. And what we found was indeed neurocan N-terminus is very nicely enriched in these perineuronal nets. However, the C-terminus has a much more speckled and diffused distribution within the extracellular space. So another thing she noticed that early in development, both fragments are close to each other. Potentially the protein is intact and not yet completely digested. But later in development, when we expect the inhibitory synapses fully formed, then the N and C terminus of the protein is completely at distinct locations. And she said maybe these two parts of the protein are functioning in a different manner. And to test that, she generated recombinant proteins of the N and C terminus and went back to her in vitro system and treated pure neuronal cultures with N or C terminus of neurocan. And to our amazement, we found that only the C terminus was sufficient to induce inhibitory synapse formation in the in-between cortical neurons. And the N-terminal is not synaptogenic. And none of these fragments impacted excitatory synapse formation. So then where is the ANCAN C-terminal located? And we were suspicious from its speckled little dotty distribution in the extracellular space that it may be localized to synapses. And she used a super resolution microscopy technique and stained the brains, wild-type brains with neurocan C-terminal antibody and antibodies against inhibitory and excitatory synapses. And what she found was that neurocan C-terminal is present in around 75% of inhibitory synapses, and interestingly, to our surprise, it's also present at 25% of excitatory synapses. So it's highly localized to synapses in the brain. Now we decided to use this to our advantage to understand which types of synapses neurocan C-terminus is localized to. And for that, we used a technique that was really optimized by our colleague at Duke, Scott Sutherland's lab, which is using proximity labeling with an enzyme called TurboID that bioaccumulates anything that is in close proximity to the bait that you introduce to the brain. And Dolores went ahead and used TurboID tagged N and C terminal of neurocan, made viruses that would express this protein specifically in astrocytes. So it's coming from astrocytes, secreted to the extracellular matrix, and then starts to bioaccumulate interacting proteins. And as a control, we use two controls. One is secreted turbo bio-ID alone, so just secreting the turbo ID. This is non-specific secretome of astrocytes. And then another one is the intracellular turbo ID, just to remove the background of intracellular biotinylated proteins. And she then used these probes and found that neurocan N and C terminal domain have really distinct interactomes. They have like a 46 overlap, but there are 166 proteins that the neurocan C terminal ELS domain interacts that the N terminal doesn't. So we said, this is interesting. This is probably the part that is most relevant to synapse formation. So what are those proteins? Don't worry, I won't go over 166 of them. However, we found something interesting in there. First of all, we found actually proteins that are localized to excitatory and inhibitory synapses as we anticipated. And a lot of really interesting proteins that we know are important for excitation inhibition balance in the brain. But one of them in particular attracted our attention, and that is somatostatin. And the reason for it is somatostatin is a neuropeptide that is secreted specifically at inhibitory synapses that are made by somatostatin positive interneurons. So we wondered if this meant that an NCAN C terminus is specifically at those synapses and is specifically controlling the somatostatin positive inhibitory synapse formation. And somatostatin interneurons form inhibition directly on the dendrites, and they are one of the major classes of inhibitory neurons. The other major class you may have heard about are the parvalbumin neurons, which form mostly connections to the cell bodies of the pyramidal neurons. And luckily, we can use somatostatin as a marker of the presynapses because it's secreted from the presynapse. And we can use synaptotagmin 2 as the presynaptic marker for the PV neurons. And now we can determine if neurocan C terminus is specifically localized to one type of the synapse or the other. And we found that it is neurocan C terminus is more enriched in somatostatin positive synapses compared to synaptotagmin positive synapses. So with this knowledge in hand, Dolores decided to specifically target only the C terminal of neurocan. And for that, she came up with a really clever idea to remove not the whole neurocan protein, but just the axons that makes those synaptogenic C terminal. And she worked again with the transgenic core at Duke and generated these mice where the exons 9 to 14 were deleted. And in these mice, you no longer have the C terminal of the neurocan, but the N terminal is completely intact and it's there. It's also going to, as expected, it is going to, N terminal is going to the perineuronal nets. But there is no C terminal, so you don't see the TL signal anymore. And these mice were very similar in terms of their inhibitory synaptogenesis phenotypes to the full neurocan knockouts. They do have reduced numbers of inhibitory synapses in the anterior cingulate cortex. Moreover, we can now use electrophysiology to start to tease out whether it is specifically the somatostatin or parvalbumin positive synapses that are affected. First, she did this by staining, and she found that indeed both types of synapses are affected, but I hope you can appreciate the somatostatin positive synapses are predominantly lost in these mice. And when we record from the layer 2-3 neurons for the inhibitory postsynaptic currents, what she found that indeed there is a reduction in the frequency of inhibitory postsynaptic currents, confirming the reduced number of inhibition. But also the synapses that are lost, the inhibitory synapses that are lost, have slow activity, and that is it correlates very well with the fact that somatostatin synapses are made onto the dendrites, so they are their firing, their firing rate will be slower than the ones that are directly connecting to the cell body, the PB neurons. So taken together, this now gives us the first molecule that we know of that is coming from astrocytes and controlling the formation of inhibitory synapses. And we know astrocyte conditioned media contains other molecules that induce the formation of other types of inhibitory synapses. So exciting future work awaits for us to figure out what are those other factors. And it shows us that astrocytes have this very complex repertoire of molecules that they provide to neurons to wire and remodel neuronal circuits and maintain the excitation inhibition balance. So many future studies, of course, remain open, but there are some ongoing experiments that we're interested in. So how do then astrocytes regulate these contradicting activities through promotion of excitatory versus inhibitory synapses? Do they provide these molecules specifically to where those sites are going to be, or are they just secreted broadly into the neuropil? And then we think that astrocytes have an ability to integrate the local synaptic environment, both the activity of synapses and also the physical presence and localization of those synapses, and modulate their activities by integrating that information and regulating some sort of balance. And we think that this is probably a fundamental property of neuronal circuits that we're just starting to unearth and identify. So with that, that brings me to my acknowledgements. Thank you very much for your time and listening to me. And of course, my wonderful lab, who has been a very big joy to work with. I highlighted the work by many alumni today, as well as by Dolores, but there are other members of my lab working on different aspects of neuronal circuit formation and how astrocytes and astrocyte microglia or astrocyte oligodendrocyte interactions are regulating these processes. And I would be happy to take your questions and discuss further with you. Thank you.
So first of all, I want to thank the gods of battery life in laptops for helping us. Cagla, that was extraordinary. And your slides are extraordinary too. They're really beautiful. Yes, we're having a little AV problems with people from the internet. So if those folks could just send us a chat, we can answer. But yeah, go ahead, Hezi.
And actually regulates the process of the synapse formation. Is it more of a guidance molecule? Is it help us take resolution once the synapse is formed? What is kind of our current understanding of that?
Yeah, thank you very much. I think that's a phenomenal question. And we have now a repertoire of synaptic molecules that we know is interacting with neurochem. And what Dolores is trying to do is to target those molecules that she identified in her bioID and determine which one might be downstream from neurochem in terms of stabilizing or promoting synaptogenesis.